Okay, a few more seconds, or it's okay.
Let's give it like 1 more minute, but it looks like our numbers have stabilized here. So just
Perfect.
yeah, maybe 1 more minute and then go ahead and get started.
I cannot hear you. Sorry.
I don't know if anyone else is speaking. So can you hear this?
Yeah, Beth, I can hear you, but I cannot hear Jessica. Unfortunately, I cannot hear you yet. But I can hear other persons, so it's pretty strange.
Yeah, hopefully Jessica will be able to fix her audio. Why don't we go ahead and get underway?
Yeah. So I think we can start. I don't know if we wait for Jessica a little bit more.
Yeah, let's go ahead and get started.
Yeah, okay. Hello everyone, I'm Mario Cruz and I'm a postdoc at Siemens Lab in the Broad Institute of MIT and Harvard, and I'm here on the behalf of the Cell Profiler team. Cell Profiler is a versatile open source software for quantification data from biological images, particularly in high throughput experiments. Using an point and click graphical user interface, you can construct an image analysis pipeline. So you can analyze and process your image through this pipeline to perform an illumination correction, object identification, and object measurements. So you can download the Cell Profiler software going to the cellprofiler.org website, and it's pretty easy and straightforward. Once downloaded and installed in your machine, you can open the Cell Profiler. And if you've never been in touch with Cell Profiler, this window will pop up and will guide you through the different types of tutorials or examples that we have. So in this first section, we will analyze the 3D nuclei samples. So to do that, inside Cell Profiler, we have here some modules that are image, metadata, names, types, and groups. And here in image, we can just drag and drop our images. You can drag and drop the entire folder of containing your image, or you can select each image do you wanna analyze. So if you double click, you are able to see your image, but because these are 3D images and here we cannot see the 3D image, we need to assign this as a 3D image. And to do that, we can just upload a pipeline, or we can go to names and type and assign this image as a 3D image. Here I will just upload my pipeline. And when I upload my pipeline, all the information that I need, it's assigned here. So we can just change this. So we have image. In this case, we didn't need to extract the metadata of our image, but we need to assign all images in as a 3D image with a relative pixel spacing X, Y, and Z. And in this case, we assign with the 0.124 microns for X and Y and 0.2 for Z. And we also assign here as a grayscale image, but you can also provide a color image or a binary mask to Cell Profiler to analyze. And this is a important point. You need to assign all these images with a name just to call these images after in the pipeline. So after doing that, we can just hit update, and then you can see all the images here. We come back here to the image and double click the image, now you can see that this image is read by Cell Profiler as a 3D image, and you can use this slider to change the planes. Here you can also magnify a little bit your image or just come back. And if you right click, you can adjust the contrast, for example, if you have different image subsets. Just to see that there are different images to play 1 to 4, and this is the most noised image. So in this pipeline, we can separate this pipeline can separate into parts. The first part is the image processing. In this case, we have 3 modules: the raise scale intensity, Gaussian filter, and reduced noise module. All these 3 modules will pre-processing our image, to then we provide a segmentation that will be done with the threshold and watershed modules, and then we can provide the measures and then convert objects to images and save images and overlay objects just to have a visual result. So the first model that we call here, it's the raise scale intensity. And if you just want to put another model here, you can just double click in this window, models, then this add models window will pop up, and then you can just write the name of the model and double click in this, and they will appear in your line. If you click with the right button, you can add something, another model, or you can delete this model or disable or duplicate this model. If you don't have about what this model can do for in your pipeline, you can just hit this question mark here, and a help for model window will pop up for you. So this rescale intensity model will change the intensity range of an image to your desired specifics. And this works in 2D and 3D in respect masks. So while you're doing this, so just to test our pipeline, I will just hit the start test mode button, and then we can test the models and the pipeline throughout our image. So if we click the step button here, we can see that the first or the original image has this kind of representation. And then when we rescale this image, we have another histogram definition. So let me just normalize this. Okay, yeah. Okay, yeah. So what this means actually, if you just look at this bottom part of this window, when I pass my mouse through the image, I can see the intensity of the pixel. And in this case, the black region or the background we are seeing here, it's 0.0008, and our signal or our nuclei are 0.0091. So the difference between the background and the signal, it's low. And because subprofiler just rescale all that from zero to 1, we can see here that we are not seeing the entire histogram of our image. So when we scale, we just change this histogram. So now our background intensity is 0.05, and our signal is 0.88, for example. And this will make easier just to apply the threshold and the segmentation models in our images. So this is what this model is doing. So here we need always to select the input image. So in this case, we select nuclei. That was the name that I gave for all these images in the names and types model. And I will just stretch each image to use the full intensity range in this case. And I will rename the output image as rescale intensity nuclei. So moving forward in the pipeline, we can see here that in this case, the Gaussian filter is doing too much. It's just blurring a little bit our image. But what's happening in the other image? So if you want to see what the model is doing in other image, or if you want to analyze other images in this test mode, you can just hit this next image set button, or you can go here to the test and then choose image set. In this case, I will choose, for example, the nuclei 3 image. Okay. And then we can just click here again. And now we can see a difference between the raw image and the Gaussian filter image. So the image is blurred, but we have less noise in this image. So this image is looks more like the no noise image. And in this case, we just select the rescale intensity nuclei. We name this output image as Gaussian filter, and here we are using a sigma as 1. If you improve the sigma, for example, for 3, what this means for the software, this will blur more your image. So in this pipeline, I'm just using 1. So you can just change the sigma value, and then hit the step button again, and this will update your image window. So because we can see here that this image is still a little bit noise, we add another model called reduce noise model. So as input image here, we are using Gaussian filter image from the last or from the previous model. And we are using a size of 5, a distance of 2, and a cutoff distance of 0.2. What does this mean? If you don't know, you can always call the question mark button. And so what's the size in this model means? The size of the patch to use for noise reduction. The distance means the maximum distance in pixel to search for patches to use for the noise, for example. So if you have any doubt or any problem during your pipeline construction, you can always hit this question mark button and see what this function means and what this is doing inside this model. So we can hit here again. And this step takes a little bit more time. So now here we can see that the image is blurred, but we can see here that it's less noise than in the first image or when we start. So these are the images just to compare the raw image with the denoised image. Once you have all images denoised, so we can just see, for example, here that even the most noised image is or looks like the same as the previous one. So they have a similar pattern or distribution pattern or spreading pattern of light. So when you have this normalized, you can just apply a threshold or a watershed or an algorithm to segment your objects. So because we normalize all images, we can just use one pipeline to segment through all the images they provide in this study. So the first step here is to convert this image. This first image is reduced noise image in a binary image. So a binary image, it's just when you convert all the black or the background as 0 and all your objects or all your elements inside as 1. So this image just has 2 informations, 1 for the white and 0 for the black portion of your image. And using this threshold image, okay, just coming back here, and how can we perform the threshold? We select the input image, we name this output image, and here are some things that you can just adapt to your pipeline, to your image, just to perform better. So you can choose, for example, a threshold strategy as global or as adaptive. The difference is how the threshold will work in our entire image that we will apply using a global setting or a partial setting or an adaptive setting. So here you can choose the threshold method. So we have different types or different algorithms or methods to segment your image. In this case, we are using Otsu with 3 classes. And why you're using this? Because we can see here that this image has, for example, one class that's almost black, another class that's gray, and another class of pixels that are almost white. That's our signal. So we have three classes of pixels here. So because of this, we choose a OTSO method with three class separation, and we are choosing here the second class or the second class as this gray class as a background. And then we can just apply, for example, a threshold smoothing scale just to smooth a little bit, or we can just, for example, put a threshold correction factor here. And this will limit the amount of the threshold you're putting your image based in the intensity of your image. So after that, we can just pick up this binary image and use another model called watershed model. And this model has two options, a default, a basic settings and advanced settings. In this pipeline, we are using just a basic settings. So we are selecting the threshold image. We are naming this image as, or these objects as watershed. And we are generating these objects from distance. And here we are need to provide the footprint and the down sample. So this is the algorithm that will segment our cells or nuclei in this case. So I just hit this step button, and now we have the segmentation. So we end up with different objects. Each color here in this image is an object that's identified by this module in this case. And you can just set up, for example, for a higher footprint. And what this means, you can just hit the question mark. So bigger footprints will suppress your single maxima. So you're gonna find less objects if you improve your footprint. So I just change here for 20. And now we can see that instead of 3 nuclei in this image, we gonna segment just 1. I think this takes a little bit to segment. And the down sample here, it's a way that we have just to down sample our image prior to the watersheding procedure. This is good because if you down sample your image, the time or the computational consuming will be lesser, so you can improve the time of your image analysis pipeline. So I just coming back here for the original footprint. Okay, after segment our objects, we found, based on the calibration image, that all this process that we did before shrink our object. We are using a dilate object model just to recover the object size or real size. So we are picking up the watershed objects from the previous model and naming these objects as real size nuclei. And we are using here a way or a shape to dilate our image in a size of 1. So we're just dilating our image a little bit. So now that we have the segmentation of our objects, we can just measure. And how we do this in Cell Profiler, it's pretty easy. You can just add a module called measure object intensity. And here it's super important that you select the original image. So here we are selecting the image nuclei. That's the original image, the raw image that we provide to the software. And because we wanna measure object intensities based in an object, we need to choose an object. In this case, we are choosing the real size nuclei. That's our real nuclei object. And then we just hit, and all these images or all these measures, sorry, will be done for the entire image or for all your objects. And here are just the mean, median, and standard deviation for all objects from each image. In this case, just for the image we set up in the image set. Then we measure the object size in shape. So here we have, sorry. So here we have features like volume center of X, Y, and Z. That's what are they asking us? The surface area and other measures related with the size and shape of our objects. In this model, they just ask us for the object that we are measuring, because in this case we are measure the size and shape of the object and not anything in our image. So we can just choose our object here, and if you want advanced features, you can just hit here, and this will provide you more information. For example, enable solid measurements for 3D image. So now we denoise, segment, and measure our objects. And so we can overlay our objects just to our original image to provide some visuals. So in this case, we have, we have the original image here, the rescale intensity nuclei. And here we have an overlay objects, so we can see how our objects that we segmented overlay with our original image. And we can do this for all the images. So we can just go back to the test tab, choose an image set, and for example, we can choose, for example, the AI2 image set. And in this case, if you wanna see here, it's a pause button that you can just click or unclick. So if I click here in the pipeline and hit run, the pipeline will run through all these models and stop here before these models of save image start. So we can just see what's happened. So here, So here we have the objects overlaid in our original image. After that, we can just save our image, and saving our image in images is a process of choosing the image and choosing the name. After choose what type of image do you want to save, if it's an image or if it's a mask or if it's a crop or if it's a movie stack, you need to select the type of image do you want to save. Then you can just pick up, for example, the file name from your original file, just to not rewrite everything again. So you can just select these from names and types, and you can, for example, append a suffix to the image file just to know what you're exporting. Select the file format as JPEG, in NPG or TIFF. You can choose your bit depth, and we have another options here. It's pretty important just to choose your output file location. So you can just click here and choose the output of the saved image as the same of your input folder, or the same as the input folder but with a subfolder, and you can just select this, or you can just choose the default output folder that you can set up here in this output settings. So if you hit this, you can just change here your output folder, and you're gonna save your images there. Then, because we cannot save objects in this save model, we have a round to that. So you can convert objects to image. So you can just, convert objects to image. So you can convert these objects that are your nuclei as a image. And in this case, I choose the real size nuclei, and I choose the color format of this output image as a grayscale, and then I can save this image as any other. So you're seeing this red button here. This is just because the export to spreadsheet will not work in the test mode. So if you just exit the test mode, this model will be green again, so you can export your measures to a spreadsheet or to a data set again. So here in this export to spreadsheet, you can just choose everything or how you export your data. And for example, here you can select the measures do you want to export. If you don't want to export every measure that we did, that are many. So in some cases, you don't need all this information, so you just can select here as yes and press this button. So here we can just go to the real size nuclei, go to the area shape, for example, and then choose here just the center of the object, X, Y, and Z, and then we can just select the volume of the object. And from intensity, we can just extract or export the integrity intensity measurement. Okay, when we do that, we just export all this information for a spreadsheet that I'm just gonna show you. So this is how the spreadsheet looks like. So an image number. So here in this case, we have 4 images, 1, 2, 3, and 4 images. Each image has 5 objects in this case. And here are the file name, the path name of each image. And here we have the area shape in X, Y, Z, the volume, and the intensity, the integrity intensity of the nuclei. Note here that all these values are in pixels. So we need to convert these values that are in pixels to microns. And how we do that, we can just use an Excel to do that, or we can use whatever software do you want just to multiply the values here. But we did a Colab notebook, and this Colab notebook will, that I will show here. So this Colab notebook will pick up the value of X in microns, Y, and Z. They will calculate the voxel size for our image, our voxel. Then we're gonna import our original CSV file to this Colab environment. And then we can read this file and multiply and extract the information that we need for the study. And the end of this notebook, we have a cleaning code just to clean the path. So first of all, we need to import the dependencies of this notebook. There are NumPy, pandas, and OS. So here, the second part of this code, we need to input our X, Y, and Z values. So 0.124 for X, 0.124 Y, 0.2 to Z, and they will print these values for us. If we hit the calculate the voxel size in cubic microns, they will calculate for us, and now we can import our CSV file from the output file from Cell Profiler. So here are my experiment, real size nuclei, .csv. That's the same file that I export here. So I can open this, and this file is now here on my files in Colab environment. I can call this file as a data frame, and this is how our file, our CSV file looks like. And then we can just insert new columns to this file, and calculate, for example, the area shape X multiplied by the X that we give here. So we can just click here, and then Colab will download a new spreadsheet or a new CSV file. And now you can see just the image number, the object number, the file name of your files. And now we have the X, Y, Z, the intensity, and the volume, all in the right scale in microns or cubic microns. After that, we can just hit this button, and what's happening here is just erasing these files from our Colab. So you just clean data. So here we can just use At the end, we can use Phoebe. That's another software just to see the results of our segmentation. Okay, let me just show you guys how the segmentation process works. And here's the output, or the 3D output of our segmentation in these images. So that's everything that I want to show you today. So if anyone has any question, please.
Yeah, I was wondering, is there a way to see how many objects were found in each image?
Yeah, actually, yeah, you can just, for example, export or see the summary data. So you can just, here in this export spreadsheet, you can just, for example, choose here, and you can just export the number of objects, for example. You can just select this measure and export this measure for you.
Hi, Mario. Can you hear me?
Yeah.
I had a question, which would be how hard would it be, or would it be possible to author a to like make your own module to do the unit conversion that you did at the end? So it would be like an import to pandas, basically the manipulations you did in the Jupyter notebook. Would it be pretty straightforward for a user to create their own custom module in Cell Profiler to tack that onto the same pipeline?
Okay, so you're asking if you can create this dependence inside Cell Profiler just to have this out in the end?
Yeah, essentially, because you load the pixel dimensions into the Cell Profiler pipeline, so in my understanding, all the data is basically on hand.
Yeah. We are discussing this the last week with Beth and all the group, and it's not an impossible thing to do. And we are just thinking maybe Beth can bring us a little more. I'm just seeing that Beth's not here anymore, but yeah, we are thinking about that. And maybe in future release of the Cell Profiler, we will have this module for sure.
Cool, thank you.
You're welcome. And besides of that, all these pipelines, the documentation, and this notebook to convert the data will be available on the Cell Profiler tutorial website. So you can just go there and download and test everything if you want. Okay, any more questions? So I think I can move forward. So yes, you can come back later to questions, no worry. I will just start the second part of this section. And for that, I will use the Cell Profiler again, for sure. I'm just opening the Cell Profiler software. So Cell Profiler software will pop up this window. Welcome to Cell Profiler. So here you can just see or try an example or building help or getting started or say see some tutorials that we have in our website. But here, dealing with our data, in this case, now we have the fish in C. elegans. And how cell profiler works, it's pretty simple. We have models just to call functions or to call something, to call some algorithm, or in this first model called image is just to call your image, so you can drop files or folders here. So you can just drag and drop your entire folder. If you have more files than just image in your folder, like this DS store, you can just exclude that and just select the image here. If you double click, you can see your image here, but this is just a 2D image. To convert this as a 3D image, you need to assign this as a 3D image here. So now if you open this image, you can see that it's a slider up here, and you can just slide on your image. Because these spots are too small, we have a zoom tool here. You can just pick up this tool and just zoom in image, or if you click in this house, you can just return to the previous scale. Okay, here we can see that there are different images, and each image has different signal to noise ratios, and this is the last one, or the fish 4 image with low signal to noise ratio. So what I'm doing here is just, after load my image in Cell Profiler, I can just drag and drop a pipeline here in this white window above these other models. So yes, I wanna pick up this. So if you don't have a pipeline yet, you can just double click in this white portion of the window, and now add models will pop up for you. So you can here just choose, for example, a Gaussian filter model. If you double click that, this will be added to your pipeline. If you don't know what this Gaussian filter does, you can just click here in this question mark, and a new window will pop up for you. So saying that the Gaussian filter will blur your image and remove noise. And the way this filter works, it's Gaussian. So here are all our models. It's more than 90. So you can just combine all these models in this pipeline to have your answer. So here I just delete this. So if you just right click inside, right click the model, you can delete this model, you can duplicate, you can disable, you can ask for help, you can trace inputs and output for this specific model, or you can add another one. When I load my pipeline, all the information that I need is provided. So names and types is provided. So here I am assigning all my images as fish. So all my images will be called fish by Cell Profiler. And here are the relative pixel spacing for X, Y, and Z. And I'm telling the software that this is a grayscale image, but we can also work with color image and binary mask image. So here we can just click this eye just to see the results of our models. disable or enable our model, or we can just start the test mode. When we start test mode, this will just give us the opportunity to see what's happened throughout the models. And we can just hit here this green button or click here in step. If we click here in step, we're gonna see that the image, the original image, will be blurred by our Gaussian filter. And this is the result image. We can again just zoom in this and see what's happened. So this pipeline has three parts, or three main parts. Again, a part just to remove the background of the worm and to reduce the noise. That's up to rescale intensity. The threshold and watershed models are the models just to segment the objects or the spots. And all these other models, from Mentor to export to spreadsheets, are models to export data or to calculate some kind of measure. In this case, object measurement. So the first part, or the pre-processing, we'll just remove the noise and remove the background of all your images, just to make the threshold and watershed process easier. And this could be applied to all images at the same time, or using the same pipeline. Sorry. So in this case, we have 4 images, so we can just, and if we click here, we're gonna see just the first image, but we can just click here in next image set. This will move forward in the image set. So this is the second image, the fish 2, or we can just go here up, test, choose image set, and then choose, for example, the fish 4, that's the lower quality image. And now we're gonna see here. In the first part of this pipeline, we'll just remove the worm, so we're not caring about the spots yet, so it's not a problem here, just to have this very blurred image. So after blur our image, we can see very clearly our worm, so we can segment our worm, so we can create a mask of our worm. And the way we do that is just using a threshold method. So the threshold method will segment, or will transform your image from a 8-bit image to a binary image. So here we can see that the pixels has values of from 1 to 0, and they can vary depending on the intensity. So we can see the intensity here in this bottom side of the window. So when I move my pointer throughout my image, I can see the value or the intensity value for each pixel. Okay, and in this new image, that's the threshold image, all the black pixels have 0 and the white ones are 1. So we have just a binary image. In this case, we are using as input image a Gaussian filter image that was provided from our first model. We are naming this image as a output image, as a threshold. We are using here a threshold strategy as global, but we can choose an adaptive strategy. If you don't know what this means, you can always go here to this question mark, hit this button, and this will explain you what's the difference or what's going on in the global or in the adaptive case, and some tips. Okay, this global method will be faster and robust. For example, if the background is relatively uniform, for example. In this case, we have, so we choose the threshold method. So again, if you don't know what this is mean, you can just go here, hit, and a new window will pop up explaining the difference or the base of each method. So after the transformation of our image, we can dilate our image a little bit. So this model will just expand the threshold values or the threshold area. So here we can see, for example, that we just dilated a little bit our image. This is important because the next step will be mask our image. What does this mean? We're gonna remove all the background outside of the worm here. So here, so we have the image. Everything outside the worm has a value of 0. That's completely black. And this is pretty good to us because we can remove this variable from our segmentation process. Then what we did is, just because we have the dots here or the spots here, but we have a lot of noise yet. So what we did is just measure the intensity of the worm. And here, when we measure the image intensity of the masked worm, we have some particular measurements about the median intensity, the median intensity, and the standard deviation intensity. And if we look to the image, let me just pick up the image again. Most part of our image or the masked worm is background or it's noise. So if we just calculate, for example, subtracting this masked image by this measure, called median intensity measure of the masked worm, we can remove most of our background and noise of our image. So that's pretty good. After that, we can just rescale our image. This will just, what rescale does here is just, we just rescale the intensity of the pixel. So here we are not seeing anything because we are using just a range of our histogram. And now in this rescale image, intensity image, we are seeing the full spectrum of our histogram. After that, we can threshold this image again, but now we are looking for the spots. So now we can just see the little spots here. So now we have 4 spots here and 4 spots were separate here in as a binary mask. And after we have a binary mask, we can apply the algorithm to separate the objects inside this pipeline. And this can take some time because the image is big, we have a lot of spots, so this could take some time to run. To this pipeline, we can run this pipeline throughout our image in 2.2 minutes and a half. So it's pretty fast. So here in the end, we can just see the spots, and each spot has a color assigned. That means that each spot is a different object here in this image. So we can just move or slice this through the planes. So after that, we can just measure the object intensity. And in this case, we need to select the image that we like to measure. And we always recommend to measure in the raw image that was provided. So the raw image was called as fish. So we're gonna select the fish image, the original image, and then we can select the object to measure that there are our spots. When we hit this button, this will pop up the means of our measurements. And here we have a lot of features, so you can choose whatever feature you're gonna export to your spreadsheet in the export to spreadsheet module in the end of this pipeline. So here I'm just using another rescale intense model just to provide a new image using the full range of our histogram, but now for array scale intense of our original image and not the masked warm image. And why I'm doing that? Because I want to overlay the lines. So here we have a model called overlay overline. So you can just select this model, add this model to your pipeline, and then you can overlay the spots just to see if you're segmenting everything you want or not in your using this pipeline. And then we can save our images as TIFF or as JPEG or PNG. We can choose the way we're gonna save our image, and most important, where are the outputs of this image. So you can just click here, output file location, choose your output folder, and can be the default input folder with a subfolder or whatever you want. Because we cannot save objects using the save image model, we can convert the object to image and then save the objects as image and see what's happening. I actually can show you the projections. Okay. So here is the projection of the 3D image of all our spots that were segmented throughout this pipeline. And now we can exit the test mode and export to spreadsheet our values. So here we can choose to select the measurements, or we can just export everything if you want. So because in this case of this study, they are just asking us for the intensity values or the integrity intensity in the location of the spots, we can just go through here and select some measurements. So we can go to spots, intensity, and just export integrity intensity. And we can go to location and export the center of each object in X, Y, and Z. And okay, when you're done with your pipeline, you can just hit the Analyze Image button, and then this will analyze all the images that you provide here in the first part of this tutorial. The output CSV file will be like that. Just show me. Okay. This is the output of our. So here we have the image number, the object number, and the file name, the X, Y, Z, and the intensity value. This the integrated intensity value of our spots. But Profiler will export this data as a pixel values and not in microns or cubic microns. So we can transform this using a pipeline. So here we just create a notebook. And what this notebook is doing is just importing some dependencies or some libraries. So we can hit here. We are gonna import some of these libraries, and then this notebook will ask us for the dimensions. So in this case, it's 0.162, if I'm not wrong, and 0.2 for Z. And then what we need to do is just to import the original CSV file from Cell Profiler. So this will be for example, output. Okay, this is uploading the file for us. Once the upload's finished, you can just hit here, the next button, and this will read our CSV file as a data frame. Once we read this, we can just make some modifications to the CSV file. And in this case, we are just transforming the X value that's in pixel, the X value, or location center X. This value is in pixel. So we are just multiplying this value by the value that we provide here for X. So this is what this function is doing. And then this function will download the new CSV file for you. So if you just click here, this will do all the operations, and then you can see the image, the object number, the file, the X, Y, Z, and the intensity value for each spot. Okay, and the last part of this notebook is just a code to clean your files. So if you hit this, will clean your notebook so you can import new files to your notebook and process this again. Okay, this is what I like to show you today. So if anyone has some question, please. Another thing that I like to comment is that all these pipelines and these notebooks from Colab are available in the tutorial section of the, will be available in the tutorial section of the Cell Profiler website or GitHub. Thank you very much for the organizers and everything.
Thanks, Mario, for that really comprehensive coverage of how to process those images. If anyone has questions at this time, please feel free to just unmute yourself and ask them. We've got, I think, about 10-ish more minutes in this room, and then we can go back to the main room, and then you can go into the other rooms. I wouldn't recommend jumping into other rooms right now because you might interrupt what's currently going on, but please feel free to ask Mario any questions about either this data set or any other.
Yes.
So I have a question. I've used Cell Profiler just very briefly in the past, and for batch processing large data sets, it asks to export to a database instead of export to a spreadsheet. But honestly, I had no idea where to even open the database after it had been created. So can you talk about the differences between those two?
Let me just see if Bet's here. No, Bet's not here. Yeah, the biggest difference here, yeah, you can just hit and make sure, let me just. So when you're working in export to database, you're dealing with a lot of data for sure. And so you need a proper program to call this file. I think here we use the MySQL software to open this database, but we sometimes do these files just to open another softwares, such as, for example, Cell Profiler Analyst, just to analyze our data. But I cannot provide you much more information about that. Sorry.
Any other questions right now?
I'm just checking the chat here, and I think we don't have anything there.
I think we covered everything. I'm going to stop the recording.